Hey guys, it's Olivia. Welcome back to your endocrinology section. In this chapter, we'll be covering some insulin physiology, and this is going to be pretty heavy on the biochemistry, so apologize for that in advance. However, the physiology of how insulin works in the body and how the secretion of insulin is regulated is highly testable content for step 1. So it'll be very high yield for you guys to be familiar with these pathways before taking the test. Before we dive into the details of how insulin is secreted and how this is regulated, let's go over some of the basics of what insulin's job is. So insulin is an anabolic peptide hormone. Anabolic meaning it's responsible for building more tissues. Think of anabolic steroids, those are responsible for building muscle, right? Versus catabolic hormones, which are responsible for breaking things down. I always think of cats as mischievous and destructive. So catabolic hormones are responsible for the breakdown of tissue, whereas anabolic hormones are responsible for building up more tissues. It is a peptide hormone, so it is a protein hormone. And in the most basic terms, insulin is going to be released when you are in the fed state, when you have surplus energy, surplus glucose, and your body says, we have a lot of building blocks, so we're going to be able to build more tissue. So it should make sense to you that insulin release is going to be regulated based on the amount of glucose and ATP in the body. If it's an anabolic hormone, we only want it around when we have the building blocks to build more tissue, and we have a surplus of glucose and ATP. So high levels of glucose and high levels of ATP are going to lead to increased insulin release. Insulin is released by our beta cells. These beta cells exist in the islets of Langerhans in your pancreas, and they are responsible for producing insulin. And then looking over here, we can see a more detailed picture of the regulation of the release of insulin from your beta cells. So we're in a pancreatic beta cell, and you can see that our glucose molecules right here, these red hexagons, are entering through our GLUT2 channel. This GLUT2 channel is allowing the glucose molecules to enter into our pancreatic beta cell. And when the glucose is inside the cell, enzymes like glucokinase are going to convert it to glucose 6-phosphate, which will then allow glucose to go through these different pathways like glycolysis, the citric acid cycle, the electron transport chain, all of which produce ATP. As more glucose enters our pancreatic beta cell in the fed state and we are producing more ATP, ATP can then subsequently cause gating of this potassium channel. And I know it's a little bit counterintuitive because you might be familiar with ATP in the setting of activating that sodium-potassium pump, but in this case, ATP is closing a potassium channel, leading to the buildup of potassium within our pancreatic beta cell, which will eventually lead to membrane depolarization. And once we have membrane depolarization, that is what is going to stimulate this calcium channel to open. So the depolarization stimulates our calcium channel to open, and is the influx of calcium into the pancreatic beta cell, which is going to stimulate the translocation of these vesicles containing insulin to the cell membrane. And as these vesicles fuse with the cell membrane, they're going to release insulin into the extracellular space. So as we just discussed, the steps for glucose release from your beta cells are glucose entering into your beta cell through the GLUT2 transporter. Once the glucose is within your cytosol of the pancreatic beta cell, enzymes like glucokinase can convert glucose into glucose 6-phosphate. Glucose 6-phosphate can go through glycolysis, the citric acid cycle, and oxidative phosphorylation, all of which are going to lead to the production of more ATP. More ATP is what is going to close our potassium channel. This will lead to a decreased potassium efflux, a buildup of potassium within the cell, which will lead to depolarization. Depolarization leads to opening of our voltage-dependent calcium channel, and this calcium influx, which is eventually going to lead to the increase in insulin secretion. So that's the release of insulin. But what happens once insulin is released? As we mentioned before, insulin is an anabolic hormone that is going to stimulate the growth and differentiation of muscle tissue, fat tissue. Zooming in over here, we see an example of one of these target cells. We can see that our insulin molecules right here are binding to a tyrosine kinase receptor. This tyrosine kinase receptor, once insulin is bound, is going to autophosphorylate, so phosphorylate itself, and in doing so, become active and activate two pathways that you should be familiar with. In the first pathway, we have PI3K, which is going to activate the conversion of PIP2 to PIP3 or PIP3. PIP3 then activates PDK1, then PDK1 goes and activates AKT or ACT. And ACT is a very important protein in this pathway that I would recommend you be aware of because ACT has several jobs. First is going to stimulate the transposition of the GLUT4 receptor into the cell membrane. So these GLUT4 receptors are just hanging out and floating around in the cytosol, but when ACT is activated, then they transpose into the cell membrane at higher rates. And then of course, you have more GLUT4 receptors available to facilitate the influx of all of this glucose in the serum into your cells. So this is helpful because it allows the cells to get these useful building blocks like glucose into the cell in order to use that molecule for energy and all of the building the cell has to do. Additionally, ACT activates other molecules like mTOR1, which then goes into the nucleus, and within the nucleus will activate several genes responsible for promoting lipid synthesis. Additionally, ACT is going to inhibit FOXO or FOXO, and in doing so, it will decrease the rate of gluconeogenesis. We don't need to perform gluconeogenesis if we have enough glucose in the serum because we don't need to build more of something that we already have in abundance. And then finally, ACT will also stimulate GSK3, which will increase glycogen synthesis and allow you to store some of that extra energy that you have lying around in the form of glucose in the more efficient, compact form of glycogen. So that's the PIP3 and ACT pathway. Another pathway that's definitely worth mentioning is the RAS-RAF-MEK pathway. So over here you can see that the same tyrosine kinase receptor can also activate SOS, which will in turn activate RAS-GTP. RAS-GTP is responsible for activating RAF, which then activates MEK, which activates ERK. And then this pathway also results in effects within the nucleus on the DNA, which are going to promote cell growth and proliferation. So to quickly run through what was going on in our adipose or striated muscle target cell here, first we have insulin, which is going to bind to your tyrosine kinase receptor. Your tyrosine kinase receptor is going to autophosphorylate, which will result in activation of several pathways, the PIK3 and ACT pathway, or another, the RAS-MAPK pathway. And you might see slightly different names of each of these pathways. Sometimes it might be the PIP3 and ACT pathway or the RAS-RAF-MAPK pathway, but they're generally referring to the same pathways. In the PIK3 or ACT pathway, first you have PIK3, which will phosphorylate PI to PIP3. This is going to recruit PDK1 and ACT. ACT is an important molecule in this pathway and definitely one that's worth remembering. PDK1 is what is going to activate ACT, and then ACT has several jobs, one of which increasing levels of mTOR within your cell. Additionally, this pathway is responsible for several other of insulin's downstream effects, increasing protein synthesis, increasing glycogen synthesis, increased translocation of GLUT4 to our cell membrane in skeletal muscle and adipose cells. This again is going to facilitate increased entrance of glucose into these cells for use. And then finally, we discussed the RAS-MAPK pathway as well. And in the RAS-MAPK pathway, we have RAS activating RAF going to MEK1, MEK2, and downstream effects of this pathway include increased growth, cell differentiation, and cell proliferation, all consistent with the end goal of insulin as an anabolic hormone to increase growth and building of new tissues. Now, I know that was a lot of information. Let's briefly recap what we talked about in this video. So insulin is made by beta cells in your pancreas. Insulin is released during a fed state when glucose levels are high and ATP levels are high. When insulin is released, it then travels to skeletal muscle and fat cells, where through either the PIK3 and ACT pathway or the RAS-MAPK pathway, it can exert these downstream anabolic effects, increasing protein synthesis, increasing glycogen synthesis, increasing cell growth, differentiation, and proliferation. And I know we covered a lot of different intermediates and biochemistry in this video, but I think if you remember at least the very basics of why insulin is secreted, what its goals are, and what its end effects are, and then maybe a couple intermediates in the pathway, that will serve you well for step 1. And how would they ask you about this on a board style question? Well, something they might ask is, a 15-year-old boy presents to the emergency department for 3 days of fever, chills, diaphoresis, nausea, and vomiting. Labs reveal a serum glucose of 680. A deficiency in which cell type is likely responsible for the underlying cause of the patient's illness. Okay, so we have a 15-year-old patient. He is in the ER with pretty nonspecific symptoms, fever, chills, sweating, nausea, and vomiting. Could be the flu, could be some sort of gastroenteritis, but we have a glucose of 680. That's incredibly high, and in a pediatric patient with this constellation of symptoms, that's pretty suspicious for diabetic ketoacidosis in the setting of type 1 diabetes. And if we're going with the fact that this patient may have DKA in the setting of type 1 diabetes, the question then asks us a deficiency in which cell type is likely responsible for the underlying cause of the patient's illness. Well, we know that in type 1 diabetes, your pancreas is unable to secrete enough insulin. And we also learned from this video that the cells in the pancreas that secrete insulin are our beta cells. So to answer this question, I would look for beta cells on the answer list as the cell that is responsible for producing insulin and it is defective or deficient in type 1 diabetes. Now I hope this video was helpful, and I'll see you next time.